Hi, and welcome to 10-Minute Neuroscience. In this installment, I'll be covering how the brain changes across the lifespan, from early neural development to older adulthood. Of course, there's a lot of variation in neural development among individuals, so I'll be describing broad developmental patterns that show up consistently, not a rigid sequence that unfolds in exactly the same way for every person. The development of the nervous system starts at around 3 weeks after conception, when part of the ectoderm, which is one of the major cell layers in the embryo, thickens to form what is called the neural plate. The neural plate is the first visible structure that will eventually give rise to the nervous system, and as development continues, this neural plate begins to change shape. A groove forms down the middle, called the neural groove, and the edges of the plate rise up on either side to form what are called neural folds. As these folds continue to move toward one another, they eventually fuse, transforming the flat neural plate into a hollow structure called the neural tube. This process is known as neurulation, and it's one of the most important events in early neural development because the neural tube is what ultimately becomes the central nervous system, or the brain and spinal cord. Most of this process is referred to as primary neurulation, which is the folding and closure of the neural plate to form the majority of the neural tube. There's also a process, however, called secondary neurulation, which contributes to the formation of the caudal, or tail end, portion of the spinal cord through a somewhat different mechanism. But a key point is that the central nervous system begins as a tube, and that tube forms very early in embryonic development. At around the same time, another important population of cells appears at the border of the neural plate. These are called neural crest cells. Neural crest cells migrate away from the developing neural tube and give rise to a wide variety of structures throughout the body. In terms of the nervous system, neural crest cells contribute to much of the peripheral nervous system, including many sensory neurons and autonomic neurons. So the neural tube gives rise to the central nervous system, while the neural crest cells contribute substantially to the peripheral nervous system. Once the neural tube is formed, it does not remain a simple uniform tube. Instead, different regions become specialized for different functions. For example, the dorsal part of the neural tube, which is situated toward the back, becomes specialized for sensory functions, while the ventral part, which is situated toward the belly, becomes specialized for motor functions. This basic organization is an important feature of nervous system development and is reflected in structures like the spinal cord and brainstem. As development continues, the rostral, or front end of the neural tube, begins to expand into a series of bulges known as brain vesicles. Initially, there are 3 primary vesicles: the prosencephalon, or forebrain, the mesencephalon, or midbrain, and the rhombencephalon, or hindbrain. These primary vesicles later subdivide into 5 secondary vesicles. The prosencephalon divides into the telencephalon and diencephalon. The telencephalon will eventually form the cerebral hemispheres, while the diencephalon gives rise to structures like the thalamus and hypothalamus. The mesencephalon remains as the midbrain. The rhombencephalon divides into the metencephalon and myelencephalon. The metencephalon develops into structures such as the pons and cerebellum, while the myelencephalon becomes the medulla oblongata. Over the rest of gestation, the neural tube will continue to transform in shape until it starts to resemble a brain and spinal cord. One of the most dramatic aspects of brain development is how much the telencephalon expands relative to the rest of the central nervous system. Early in development, the brain is much more dominated by structures that will become the brainstem and other lower regions. But over time, the telencephalon grows at a disproportionate rate, especially in humans. This growth leads to the formation of large cerebral hemispheres, which come to dominate the adult brain. Additionally, the cerebral cortex grows dramatically, and because the amount of cortical surface area increases faster than the available space in the skull, the cortex begins to fold. These folds form the ridges and grooves, or gyri and sulci, of the mature brain. Early in gestation, the cortical surface is relatively smooth, but as development progresses, folding becomes increasingly pronounced. This allows a large amount of cortical tissue to fit into a limited volume, which is one of the reasons the human brain can support such complex functions. So far, I've been focusing mostly on visible features of development, but a great deal of neural development involves events happening at the cellular level. One of the first of these is neurogenesis, which is the production of new neurons. During prenatal development, enormous numbers of neurons are generated, and the vast majority of our neurons overall are produced before birth. Some research suggests that neurogenesis may continue after birth in a limited way in certain brain regions, but there's ongoing debate about how much of this occurs in adult humans and what functional significance it has. So the overall contribution of postnatal neurogenesis appears to be much smaller than the massive wave of neuron production that takes place during prenatal development. After neurons are generated, many of them have to travel to the right place. This is referred to as cell migration. In the developing brain, neurons are often born near ventricles and then migrate outward toward the cortical surface. Many of them do that by moving along glial cells called radial glia, which act like scaffolding. This migration is essential because a neuron's location strongly influences what kind of role it will play in a circuit. Once neurons reach their destinations, they begin to differentiate. Differentiation means they become specialized. They develop distinct shapes, express particular neurotransmitters and receptor proteins, and begin to take on the functional identity that will characterize them in the mature nervous system. For example, some will become excitatory projection neurons, and others will become inhibitory interneurons, and so on. They also begin to form axons and dendrites, which are necessary for communication with other cells. So differentiation is the process by which a newly generated neuron becomes a particular kind of neuron with a specific role to play. After differentiation comes synaptogenesis, which is the formation of synapses between neurons. During development, the brain forms huge numbers of synaptic connections. In fact, it often forms more connections than it will retain long term. This overproduction is a normal part of development. It gives the brain a great deal of flexibility, allowing activity and experience to help shape which connections are preserved and which are eliminated. So rather than constructing a perfectly optimized network from the beginning, the brain initially creates a very rich network of possible connections and then refines that network over time. That refinement involves another key developmental process, which is neuronal cell death. Although it may sound like a bad thing, the death of many neurons during development is a normal and necessary part of nervous system formation. The developing brain typically produces more neurons than it ultimately needs, and neurons that fail to establish appropriate connections or receive sufficient survival signals may undergo programmed cell death, also known as apoptosis. This helps sculpt neural circuits so that they're properly matched to their targets and don't contain cells that aren't contributing productively to the system. Synapses also undergo major rearrangement during development. Some synapses are strengthened and stabilized because they're repeatedly used, while others are weakened and eventually eliminated. This is often referred to as synaptic pruning. This process is influenced strongly by neural activity and experience. So development is not just a story of growth, it's also a story of selective removal. The brain becomes more efficient not simply by adding more and more connections, but by refining those connections and reducing redundancy. Taken together, neurogenesis, cell migration, differentiation, synaptogenesis, neuronal cell death, and synaptic rearrangement are some of the major stages of neural development. They overlap considerably, and in many cases, one process influences another, but collectively, they help explain how the nervous system goes from a relatively simple embryonic structure to a highly organized and functional brain. Although a large amount of neural development occurs before birth and in early childhood, brain maturation continues for decades. Adolescence, for example, is a particularly important period of continued development. During the adolescent and teen years, the brain is still undergoing significant remodeling. Synaptic pruning continues in a number of regions, and patterns of connectivity continue to be refined. Myelination, the process of wrapping axons in an insulating layer called myelin, also continues through adolescence and into early adulthood, and in some brain regions even extends into the third decade of life. Myelin helps neural signals travel more quickly and efficiently, so increases in myelination improve the speed and reliability of communication between brain regions. As a result, cognitive processes like attention, working memory, and decision-making generally become more efficient over time. At the same time, increasing myelination can make neural circuits more stable and less easily changed, which may contribute to a gradual shift from the flexible, highly plastic brain of childhood to a more specialized and efficient adult brain. Importantly, different brain regions mature on different timelines. In general, systems involved in emotion, reward sensitivity, and motivation can become highly active during adolescence, while parts of the prefrontal cortex involved in impulse control, planning, and long-term decision-making continue to mature later. Because myelination is still ongoing in these prefrontal regions, the connections that support top-down control are not yet operating at full efficiency in adolescence. This mismatch is one factor that may contribute to some stereotypical aspects of teen behavior, like increased novelty seeking, heightened sensitivity to peers, and a greater tendency toward risk-taking in some situations. As we move to older adulthood, the brain continues to change, but the pattern of change is different from what we see earlier in life. Aging is commonly associated with reductions in gray matter volume in some regions of the brain, and decreases in dendritic arborization, meaning less extensive branching of dendrites in certain neurons. A reduction in the amount of white matter may also occur with age, which can affect how efficiently different brain regions communicate with one another. These structural changes are often linked to declines in certain aspects of cognition, particularly processing speed, working memory, and some other types of memory. At the same time, the effects of aging are not uniform across all types of cognition. Some abilities like vocabulary, general knowledge, and skills built through experience are often maintained relatively well and may even improve into later adulthood. There's also a great deal of individual variability in how the brain ages. So things like physical health, cognitive engagement, social interaction, sleep, and exercise can all influence the extent to which age-related brain changes affect cognition. So while aging is often associated with some decline, it doesn't affect all people or all mental abilities in the same way. So although we often talk about development as something that happens before birth or in infancy or childhood, the reality is that the brain is changing across the entire lifespan. The nature of those changes differs depending on the stage of life, but the central idea remains the same. The nervous system is dynamic. It's built, refined, reorganized, and gradually altered over time in ways that reflect both biology and experience. And that's a brief overview of how the brain changes over the lifespan. So if you have any questions, feel free to drop them in the comments below and I'll do my best to answer them.